and welcome to Fundamentals of Molecular Biology. This topic will be dealing with it as our start-off topic for the virtual molecular bioscience training lecture series for African scientists, um, which is part of the integrated molecular biotechnology training for climate resilience as agriculture, co-delivered by Okpara University of Agriculture, Umudike, supported by Mastercard Foundation and the University of Cambridge Climate Resilience Sustainability Funds. I'm Professor Ifeoma Irene Ijeh. I'm the immediate past director of the Center for Molecular Biosciences and Biotechnology, Okpara University of Agriculture, Umudike. And I'll be taking you through the basic foundations of molecular biology. I must thank Professor Lainke Ibiwumi Nwachukwu and Dr. Carol Ibe for inviting me to deliver this lecture. And I welcome all participants and do hope we'll have a good time in this course. So what is molecular biology? It is the discipline that explains life at its most fundamental level, the level of molecules and informations. How are biological instructions stored, interpreted, regulated, and transmitted within living cells through coordinated actions of DNA, ribonucleic acid, and proteins. And this we'll be looking at as the molecular basis of life. All living things from microorganisms, bacteria, to trees, to elephants, to anything that has life in it are based on a universal genetic code. All organisms store the complex information they need to live, grow, and reproduce in a small molecule called the DNA. And it is written as a code in the DNA. So the central dogma of molecular biology tells us that genetic information is stored in the DNA, transcribed to RNA, and translated to proteins, which are the mediums of gene expression. And so you look like your father because the information stored in his genes have been passed from one generation to another and then to you. So at its core, it is the study of how biological information flows inside living systems and how that information becomes structure, function, and identity. So every living organism, from a bacterium to a human being, is built from the same molecular components, yet we all display extraordinary diversity, even among human beings. We display diversities in size, shape, complexity, and all that. And the secret is in the information that has been wired in our DNA. So how does genetic information flow? Like I said, these informations are stored in sequence of bases in a nucleic acid. So just like proteins are built from amino acids, fats are built from fatty acids, our carbohydrates are built from basic units, small units of glycosides, of glucose and other monosaccharides. DNA is built from amino acids, and there are long linear polymers called nucleic acids that carry information in a form that can be passed from one generation to the next. The DNA carries the message, stores the information, while the RNA is the messenger. So the RNA acts not only as messenger but as regulator, while the proteins express the message. So they perform nearly all cellular functions. So together, these molecules form the molecular logic that governs cellular behavior, inheritance, development, evolution, determining who organisms are, how their cells function, and why diseases arise. So the DNA was the structure was discovered by two scientists, James and Watson Crick, in 1953, and the two shared a Nobel Prize for this discovery. And so building on the insights that the DNA is a double helix, they articulated the central dogma of molecular biology, describing the directional flow of biological information from DNA to RNA to protein. This was published by Francis Crick in 1958. This framework demonstrates that life is not governed by vague vital forces, but by ordered molecular interactions governed by chemistry and physics principles. So what are the foundations of molecular biology? In simple terms, molecular biology is the science of the biological code. The DNA encodes genetic information using a universal four-letter nucleotide alphabet: adenine, thymine, cytosine, and guanine, arranged in linear sequences that function as a molecular script. So these linear sequences we find in what is called the genetic code, where these nucleotides are arranged in triplets of nucleotides that we call the triplet code. And the sequences of these codes are faithfully copied during replication when cells divide. So for genes to be expressed, then they whatever information is in the DNA must be transcribed to an RNA called messenger RNA. The messenger RNA then carries this information from the nucleus where it is to the cytosol where protein synthesis occurs. And this process, the process of translating the information is carried is called translation and or protein synthesis. So proteins synthesize, express the information stored in the gene. So they may eventually express the information as enzymes, receptors, structural elements, as skin proteins, as signaling molecules, as regulators of cellular processes, as hormones, name it. These proteins will then express whatever has been encoded. So replication, like I said, is the copying of the DNA for cell division. Transcription makes RNA from DNA, while translation makes polypeptides from the information stored in the RNA molecule. Now, so all life forms basically are unified by these codons carried by the DNA molecule. So molecular biology reveals how astonishing biological complexity arises from molecular precision, the arrangement of the codes of the nucleotides in the sequences we call the codons that are programmed in each DNA molecule. So, and this is so important because a small change in the codon can result in a marked effect, as we see in the case of sickle cell hemoglobin, there's the sickle hemoglobin and hemoglobin C, where just a change from an A to a T changes the amino acid to be stored in the protein from glutamate to valine or from glutamate to lysine. And the marked physiological change that occurs in the properties of the hemoglobin is results in the diseases we see associated with. So the molecular logic of life thus explains to us how one fertilized egg develops into complex multicellular organism and how genetically identical cells can adopt distinct identities because of what is programmed in them. So now, there is also a relationship between structure, function, and molecular organization. The differences in the organization of the DNA in eukaryotic organisms, that is higher organisms like us, and prokaryotic organisms, also result in a marked difference in how they operate. Like in the eukaryote, the DNA is linear, packaged around histone proteins into the chromosomes and assembled into the chromatin enclosed within the nucleus, while in prokaryotes there is no nuclear membrane. And because there is no nuclear membrane, the DNA is typically circular, highly supercoiled, and localized within a nucleoid region lacking a nuclear membrane. And the patterns of expression of the genes vary because of this molecular organization. So these architectural differences uh, are not merely structural, they shape the gene regulation, they shape how it is replicated, and they also shape the um, environmental changes beyond its conceptual power. Molecular biology is profoundly practical and underpins modern medicine. So today we know that molecular biology has brought a large transformation to molecular diagnostics, vaccine development, cancer genomics, and gene-based therapies, as exemplified in the speed with which the RNA vaccine used for the COVID-19 was developed. So what are the applications? The functional differences uh, find applications in agriculture, molecular breeding, genetic improvement, strengthening of public health through rapid pathogen detection. You don't have to do the guesswork doing culturing and reculturing because you can easily isolate the DNA from the organisms. And because today we already have the DNA sequences of different organisms, we can easily identify an organism specifically much more than we can do within it. Today we have fuels developed using recombinant DNA technology, techniques such as polymerase chain reaction, DNA sequencing, genome editing has transformed um, the biological sciences into a quantitative, predictive, and integrative science. So we have a chemical language then in the code, okay, on the DNA, which keeps a record that can be, transferred from one generation to another, a historical record of evolution process of human generations, and every biological process um, is an expression of this encoded information. So molecular biology explains how life works, from nucleotide to the organism, from sequence to function, from molecular mechanism to real-world impact. And it unravels life not as a metaphor or mystery, but as structure, sequence, mechanism, and meaning at the deepest level biology can reach. So it provides the logical explanation of why, and how we are who we are. So I'll quickly just, in a short, quickly talk about the structure of the DNA because you'll be dealing with the DNA. So it is the molecule that runs the earth, and the DNA symbolizes life and executes life. So it's an instructional architecture. If it is removed from a cell, that cell will cease to live. It will lose its identity. It will lose its functionality. it will lose its architecture. So it is the structural architecture of biology. So the DNA structurally has three major components. You know, like I said, it's made up of repeating units of nucleotides. And these units of nucleotide determine both the stability, the copy ability, the mutability of the DNA. So at the basic core, every DNA will have DNA will have a nitrogenous base, a 2-deoxyribose sugar which forms the structural frame, and a phosphate group that forms the rivet that connects the backbone of the DNA molecule. So on this slide, you see the um, structure of the nucleotide triphosphate, where you have a deoxyribose sugar, then the base is attached at the position 1, and then at the position 5, we have the phosphate moiety. So, and then the two nitrogenous, the 5 nitrogenous bases, where you have adenine and guanine, which are purines, and cytosine, uracil, and thymine, which are pyrimidines. You remember that from your undergraduate work. So these nitrogenous bases are arranged, like I said, in triplet codons. At first glance, they look so small, but these 4 symbols encode the complexity of life. The answer lies not in the number of symbols, but how precisely they interact. Like I mentioned, their arrangement into triplet codons, each triplet is coding for an amino acid. And you remember that amino acids are the basic building blocks of proteins. So they are written like the letter of life for each programming for each protein along the DNA molecule. So each amino acid that is coded for can be used in the synthesis of a protein. So every DNA has a sequence of these codons coding for each protein that will be synthesized in our body to express one gene or the other. Now, the sizes also matter. The bindings are specific. A and G, like I said, adenine and guanine, like I said, are purines. They have 2 rings, so they are bigger than the pyrimidines that have 1 ring. And this size difference is critical because the pairing, one large base will always pair with a small base. So adenine will always pair with thymine, and guanine will always pair with cytosine in the forming of the DNA double helical structure. So usually DNA polymerases will that catalyze the replication process physically measure the base geometry. So usually after replication, they must go back and proofread. A DNA polymerase proofreads to ensure that the base pairing has occurred in the right geometry, and adenine pairing to thymine, guanine pairing to cytosine, where it has been wrongly done in the process of replication, it is corrected by the specific enzyme. So, like you have also seen, deoxyribose has 1 missing oxygen, and that changes its biology, making it more stable than the RNA. That is why nature has chosen the DNA as the molecule to store genetic information because of its stability, not the RNA. So our genome, the information carried in our DNA, persists from birth to death, and even after death, if any part of our body is picked up, that information, our DNA can still be extracted and picked up. That's why you can pick bones of people who have died long ago and start tracing their origin because we can still sequence their DNA. So the phosphate groups, like I said, form the backbone that holds um, time together. They hold together the basic units the nucleotides in the DNA, and it's arranged in a stack as I've shown in this slide. So you can see from the 5' end, so it's arranged in 5 to 3' end, where the phosphate groups, or the hydroxy on the position 3 of the deoxyribose attaches to attaches to the the methyl group on the 5' position. Okay, so these bonds are covalent and they are extremely stable and resistant to heat, which is why it's been chosen by nature. So in the laboratory, it also has implications when we run electrophoresis, because the DNA is negatively charged if we move in the direction of the positive electrode. And also the sizes will differ. The strands, like I mentioned earlier, are directional. They move 5 to 3' end of the molecule. And this is important because replication, in which the copying of the DNA strand also moves in the 5 to 3' direction, and PCR primers must also bind in that orientation. Now, the DNA is a double helix, as explained by Francis Crick and James Watson. So, like I said, because adenine pairs to thymine with double hydrogen bonds, guanine pairs with cytosine. Okay, the hydrogen bonds are not the main stabilizers, because the strength comes from the stacking, and the way the stacking occurs creates the double helical structure of the DNA. So, um, the major functions of the DNA are to direct its own replication um, during cell division, also direct transcription of complementary molecules during the making of proteins, when the information carried in the DNA must be translated to the RNA. Um, I'd mentioned earlier the genetic code. I'll quickly just talk about it in passing. There are 64 codons, like I said. The nucleotides are arranged, organized in triplet codes, each code coding to an amino acid. But remember that we have just 20 protein amino acids. So each nucleotide has 4 possible combinations, and um, more than one triplet code can code for some amino acids. Some amino acids have up to 4 possible codons that can yield them, while some have 2, and so on and so forth. And this is beneficial in life, because when there's a mutation, the mutation may just cause the addition, or cause the change of a nucleotide to another nucleotide that codes for that amino acid, and the mutation will be silent. We will never know it happened, or we live our normal life. It doesn't lead to any cancer. So, but a typical example of a mutation that can cause trouble is the single base substitution, where A is substituted for T in the mutation leading to the beta globin chain, position 6 of the beta globin chain of hemoglobin, resulting in a hemoglobin with completely altered structure, red blood cell sickling, systemic disease, just one substitution of one nucleotide. Okay, uh, the gene organization also is important. The DNA, the organization of the codons on the DNA also help both in regulation and also in the expression. In eukaryotic genes, we have both introns and exons. The exon regions are coding, the introns are non-coding. So after transcription, the introns must be removed before translation of the codes on the DNA to get the right protein. So and then, like you learned in biology, the genes are located on chromosomes at specific loci. So you often will hear this gene loci. So today we know almost all the loci for the various genes programming for the various proteins, the enzymes, the hormones, and that has helped immensely in both medicine, agriculture, pharmacy, and in environmental um, biology, where we can now wire organisms to make them either clean up oil pollution or produce more certain molecules for us. So the promoter region is the DNA sequence that recruits RNA polymerases during replication. Then have the transcription sites and various sites that we will study later. So prokaryotic genes are arranged differently. They have less complexity. They do not have introns, and so their regulation is distinctly different from the regulation of eukaryotic genes, which is more complex. Most of them use operons, and these operons arrange genes programming for proteins that do related work for the organism in what is called an operon. This helps to separate, helps to save the organism from wasting transcriptional energy, regulatory complexity, time delay that occurs in eukaryotes, and also speeds up their survival in unfavorable environments. This information is used as a target in selecting antibiotics and molecules that we can use, especially in um, dealing with infections and diseases, or also in our industrial production of products by rewiring these organisms. So, like I said, the eukaryotic gene is more complex, and the organization, it has introns, it has exons, it has intervening sequences. The introns must be spliced, allow alternative splicing, helping us to produce multiple proteins from one gene, and gives regulatory flexibility also. So, from what we've said, for information stored in the genes to be passed on, there must be replication, and then for it to be expressed, there must be transcription and translation, okay, and for the proteins to be produced. So, and that's the central dogma of molecular biology, that information is stored in the DNA, transcribed to the RNA, translated to protein. These exceptions, of course, occur in retroviruses and prions, which are wired to carry information from RNA back to DNA, the process that is called reverse transcription, catalyzed by the enzyme reverse transcriptase, which these organisms express. So what happens when something goes wrong? When there's a mutation, the DNA sequence is changed for one reason or the other. We're exposed to ultraviolet rays, we're exposed to chemical mutagens, where people smoke, people eat a lot of barbecue products with a lot of molecules that can cause mutations, changing either a codon or causing various degrees of damage to the DNA. When this happens, either the proteins to be produced are completely changed, or they cannot even be produced at all, and this can lead to various diseases. So what are the techniques used in molecular biology? A number of important techniques come into use. First, and the most basic, which I'm sure you will do your hands on, is genomic DNA isolation. This involves using physical or chemical methods to separate DNA from a sample. It is not rocket science. I've done a lot of practicals for secondary school students using simple techniques where they can separate DNA from banana, from fruits, and when they see the fruit DNA, they get quite excited. But in this course, we will be using heat methods. So there are a number of methods that can be used. The conventional methods, what we call the boiling method, used mainly to extract DNA from plants and bacteria, things with very strong cell wall, and the heat method which can be applied across all forms of life. So, and there are four steps involved in DNA extraction. There must be cell disruption. The cell must be lysed because the DNA is in the nucleus, and the nucleus is right inside the cell, so we must have access to the cell. If you're going to enter into anybody's house, you must open the door, or window. You must have an access. So the membrane is like a door shielding the cellular contents, and that membrane must be disrupted or lysed for access to the nucleus where the DNA is. And then you must have a technique for precipitating out the DNA, and then for purification, for elution, and for recovery. So, and these techniques uh, apply include organic extraction, non-organic methods for extraction, absorption methods. They vary from one technique to another, and you'll be using um, mostly the non-organic method, which we use proteinase K to disrupt the membrane. So there are also basic cell disruption techniques that can be used even um, even if your lab is not sophisticated. Precipitation usually will use salt and alcohol to precipitate the DNA out of aqueous solution. As you can see in this slide, you can see they extracted the DNA after cell disruption, and then by the time we add either ethanol or isopropanol, depending on the alcohol we are using, you can see the DNA buffy coat looking off-white here in this slide. Then the third step usually is that when the DNA comes, it comes with some proteins, comes with some RNA, so it must be purified from the and separated from these unwanted debris. And then if you are doing something, you may want to assess the quality and yield of your DNA. That can be done spectrophotometrically by measuring absorption peak at 260 nanometer, or using the ratio of absorption of your extracted DNA at 260 and 280. And if you have a ratio of less than 1.7, then it means that there's protein contamination and you need to repurify. Other tools used are polymerase chain reaction. The PCR is a molecular biology tool or technique that is used to amplify a target segment of a template of DNA. Let's assume that there's a crime scene, and the person who was robbed or raped, all he could get was a strand of hair from the victim. That strand of hair will not give us sufficient DNA, but we can extract the DNA from it, and for us to be able to study it further and cross-match with all the people who have been accused, we will need to make several copies of that DNA. So the DNA, the PCR is like a copy machine which makes copies of the DNA for us. And usually, but unlike replication, in which the entire DNA is copied, in polymerase chain reaction, we amplify a segment of the DNA that we are looking for, carrying the particular gene that we want to study or analyze or confirm. So usually short oligonucleotides are used in PCR called primers or PCR markers. So this PCR was invented in 1983 by a man called Karimulis while he was working as a chemist, at the Cestus Incorporation, a biotechnology firm in Emeryville, California, USA. And what they did um, today has transformed molecular biology. So it selectively amplifies specific segments of the DNA. So it actually mimics the natural process of replication, but performs it outside the cell in vitro under controlled conditions. And the invention, like I said, has revolutionized DNA. So how does it occur? It occurs in repeated cycles where the target region is exponentially amplified while the non-target DNA regions remain largely unchanged. So each cycle doubles the target DNA, leading to exponential amplification. And PCR machines are often called thermal cyclers because of the use of heat and also the running of repeated cycles. So what's the core principle? It's complementary base pairing, DNA replication by DNA polymerase, and sequence specificity defined by primers that you are using. So your primer will determine the sequence that you are amplifying. And there are different types of PCR. You have the real-time PCR, you have conventional, you have reverse transcriptase PCR, multiplex PCR, nested PCR, allele-specific PCR, hot start PCR, etc. And the PCR markers and also differ. You have the random amplified polymorphic DNA, restriction of fragment length polymorphism, amplified fragment length polymorphism, single nucleotide polymorphism, um, the simple sequence repeats, which are microsatellite markers. So all these I'm not sure you cover in this basic training that we are doing. But what are the requirements? We need a template DNA. That is the sequence that contains the target region of the DNA we want to amplify. And then usually we'll get this template DNA from the source that we want to amplify. If we get back to my story about the hair strand, we would have got the template DNA from there. Then you need primers, which are short sequence specific DNA nucleotides that define the start and end of the amplification. Usually you synthesize these primers based on your vague knowledge of your template DNA. And then you need a DNA polymerase. Now, because the process uses heat, the tag polymerase, a heat stable polymerase, is usually used. And then you remember that the DNA is built from nucleotides. So you need deoxynucleotide triphosphates, which must have your adenine, your thymine, okay, and the basic nucleotides that you want to build. Then you need a PCR buffer. You always remember that once you're dealing with phosphate molecules, you will need magnesium ions for enzymatic activity, and then you need a nuclease-free water, because if you have the nuclease, it will be destroying the nucleotides that you're trying to assemble. So then you must have a target sequence, that is the sequence within the DNA templates which you want to amplify, like I said, and then the primers that are single-stranded, usually 8 to 25 nucleotides, which match the sequences at the ends or within the target DNA that you want to make more copies of, and they are required to start the synthesis. So what are the steps? The steps are usually 3, and then repeated cycles of this step occur. First, you remember that the DNA is a double helix. So your DNA which you have extracted from your target will be a double-stranded DNA. So you need to open it up because what you will be extending will be the single strand. So heat is used. In replication, the enzyme helicase opens the strands, but in this case, heat it's denatured at 92 to 95 degrees centigrade to break the bonds holding the strands together. And then the next step is the annealing of your primers, which will begin the extension. So the primers are annealed, then there's the extension of the double-stranded DNA molecules at a lower, at a slightly increased temperature. Then usually, but the machine is called a thermal cycler because the cycles are repeated and each cycle doubles the DNA strands. So this kind of illustrates the PCR process. So the double-stranded DNA is using heat is opened up. So you have a DNA template strand, and then the primers must anneal to the DNA template strand, and then nucleotides need to be added in the sequence of the DNA you are making the DNA grow longer and longer till you get the length that you are going for. So that denaturation, like I said, unwinds it. Then the annealing step occurs. I'm sorry about this slide. Then extension. So what are the applications? The applications in genomics research, DNA sequencing. PCR is applied in DNA sequencing, genome mapping, single nucleotide polymorphism, genotyping, phylogenetic analysis, where you build relationship trees between DNA from various organisms, population genetics. So in genomics, PCR bridges raw sequence data and biological interpretation. So it's applied in forensics. You can take DNA from blood, hair, saliva, anything, tissue. It enables individual identification, paternity testing, crime scene analysis, and so on and so forth. In conservation, it helps to study genetic diversity, population structure, species identification using environmental DNA. So in summary, molecular biology is the branch of biology that explains life at its most fundamental level by studying how biological information is stored, expressed, and regulated within cells. Focuses primarily on the interactions among DNA, RNA, and proteins, which together govern cellular structure, function, inheritance, and evolution. It serves as the stable genetic archive that encodes instructions in a universal nucleotide language shared by all living organisms. RNA, on the other hand, acts both as a messenger and a regulator, transmitting genetic information, fine-tuning gene expression, while proteins are the functional molecules by which that express the information stored in the genes and carry out nearly all cellular activities from metabolism to signaling to structure and defense. So in effect, the central dogma of molecular biology, which we have studied today, describes the directional flow of genetic information from DNA to RNA to protein. And we have also said that this gene expression is tightly regulated by molecular mechanisms, and that small molecular changes such as mutation, epigenetic alterations can have profound effects on gene expression, including disease and evolutionary adaptation. So molecular biology underpins modern medicine, biotechnology, agriculture, molecular diagnostics, pharmaceuticals, through tools such as PCR sequencing and genome editing, and by revealing how information becomes function at the molecular level and provides conceptual foundation for understanding life in health, disease, and evolution. Thank you for listening. If there are questions, we'll take them in the course of our Q&A segment. And I also believe that by the time you get onto the hands-on, you'll have better understanding. Once more, thank you, the organizers, for. So you have a few references that you can look at